Today we are delighted to have with us Professor Michele Guindani from UC Irvine. I believe some of us at least here know him very well. Michele is a professor at the Department of Statistics at UC Irvine. He got his PhD from University of Bocconi in Italy, and then spent a few years at our own MD Anderson and also at the University of New Mexico. He has a long and distinguished profile with lots and lots of interesting publications in lots and lots of journals, high impact journals, and also a few book chapters as well. He was a fellow of he's a fellow of the ASA, and he was also the editor-in-chief of Bayesian analysis for the last 3 years. So hopefully during that time he accepted some of your papers. And he's also the current AD of biometrics and econometrics and statistics.
I'm impressed.
His main research interests are in Bayesian methodology, including non-parametrics, Bayesian non-parametrics in particular, with applications in neuroimaging data, particularly Fmri data, and integrated genomics and microbiome data. And today he's going to tell us about some of his recent interesting work on Bayesian VAR models and their applications in Fmri. So please join me in welcoming Michele.
Thank you, Abhishek. You're the best chair of the seminars. I couldn't do anything like that. It's such a great introduction. Thank you. So today, first of all, I'm very happy to be here and to be here with lots of friends. Please stop me anytime if you have any questions. I like my talks to be interactive. Okay, you have to tell me the time because we think, no, it is working. Stop me anytime. I like to have questions, especially from students. That gives me the possibility to adapt the presentation and, sort of chat about my interest with you. This is how I see generally my talks, just chats. So as Abhishek was saying, this is one of the latest project I've been working on in neuroimaging, and first I want to acknowledge the help of a grant that recently finished during this work, but also more importantly the work of my collaborators, in particular Wei Zhang, who is right now a postdoctoral fellow at the University of Italian Switzerland. Then Sonia Petrone, who is a professor at Bocconi University, and Ivor Criben, who is a professor at University of Alberta, Canada. Without them, nothing of this that I'm going to present was going to be possible. So today I'm going to discuss, as Abhishek mentioned, one of my latest interests has been in neuroimaging. And in neuroimaging data analysis, one may encounter different types of data. One common type of data is Fmri data. These data are quite interesting for different reasons. So how actually are they collected? First, what happens is that you have a subject that's put into a scanner, who is put into a scanner. While the subject is in the scanner, what happens is that typically is presented with some task, or maybe just asked to lay down and think of whatever, right? But typically there is a task associated with an Fmri experiment. The task may be of different type. For example, the subject may be asked to look at different pictures on the screen of different type. The task may be to hear a sound and push a button when the sound is heard, or like we will see later, the task may be to read words on a screen from a chapter of Harry Potter, right? This type of experiments. While these tasks are performed, the magnet Fmri scanner basically records the blood oxygenation flow in the different areas of the regions as a response to the task. So what you get at the end is an indirect measure of brain activity measured by the changes in blood flow. And what we get is a sequence of Fmri scans that capture the entire brain volume every 2 seconds. For each voxel or location in the brain, what we get is a time series, a recorded measurements of the blood oxygenation level dependent board response measurements in the brain. But again, what we are capturing is just, if you want, not directly the neuronal activity, but just the blood oxygenation in the brain, in the different brain regions. This is important for many different points, especially in terms of interpretation. When we look at the goals of fMRI experiments, typically two main goals are presented. One is the goal of segregation, studying segregation of activity, of brain activity. This is also called activation. Basically, how different brain regions react differently to the stimulus. The other big topic right now in the study of fMRI experiments is the study of connectivity, which is what we're going to discuss today. And basically, the idea here is to try to understand how different brain regions work together with others. So trying to study networks, if you want, of regions working together. Then, just as again, as an introduction, there are different types of connectivities or concepts of connectivities in brain imaging. The first one is the study of structural connectivity. This is based on MRI scans, and what we are looking at is essentially the physical connections between brain regions. Physical connections, for example, captured by white matter tracts. The other two concepts relate to the functional aspect of the experiment more than the brain configurations themselves, and those are the ones captured by fMRI data. The functional connectivity, in particular, refers to the undirected statistical dependencies between brain regions. Think about correlation of time series. If you've taken a time series class, this is the type of correlation that typically you study, right? For simple multivariate time series analysis. The other concept is the concept of effective connectivity. Here, the interest is trying to understand how what happens in one region affects what happens in other regions. So it's, if you want, actually more appropriately, a concept that explains the direct influence on one regions versus others. And this is indeed what we're going to focus on today. There's been a lot of approaches to effective connectivity inference. And these are of different type. One, for example, is the study of development of dynamic causal models that are based on biological assumptions. For example, you can think in this regard like systems of differential equations, and based on biological assumptions, and then studying the parameters of these system differential equations. Structural equation modeling, again, requires specification of some known covariance structures. Bayesian networks are also quite used, although they are not completely appropriate, because Bayesian networks typically are based on conditional dependencies through the specification of directed acyclic graph. Of course, what happens is that one region may affect another region, but there may be a feedback mechanism. So an acyclic graph doesn't work in this case, right? You can expect that as one region affects another region may affect this region as well. So what we're going to concentrate and focus on is the big class of vector autoregressive models that have been used a lot also in econometrics. And here the idea is that the vector autoregressive modeling framework allows us to capture what goes under the name of Granger causality. When I talk about causality here, please forget everything you know from causal inference. It's not exactly the same thing. Here is just a time dependence that is has been called like that historically in the literature, right? But there is indeed some work that has to be done in the neurosciences to include, I think, the current work that we have been doing in statistics in causal inference. Here is still a very rough idea that takes into the fact that the causes precede and help predict the effects, right? What happens at time in one region at time t is going to be associated with what happens in other regions at time t+1. So indeed, we are just essentially describing partial correlations. So there have been a lot of Bayesian approaches with vector addressing models, especially from the group of Raquel Prado, Hernando Ombao, and Santa Cruz, and also from some of my colleagues, my students, and so on, right? So let me just focus right now on a single subject, vector autoregressive model for effective connectivity. What would that entail? And I'm just going to focus today on the inference on a single subject. We can discuss at the end about multi-subject inference. But basically the idea here is we have the measurements, what we call YT. These are the measurements on n regions of the brain. And we have these vectoral measurements in the different n regions at each time during the course of an experiment. So this vector YT is related, is put in we're going to study the association between what we see at time t in vector in these measurements with what happens at time lags before t. So the attention here from the point of view of inference in trying to study the relationship between what happens in a region at time t-1, for example, and what happens in other regions at time t, is through the matrices A1, A2, AP. These are matrix coefficients that captures the directed influence of one region versus the other. Just to provide an example, so here we consider AJ, the matrix at lag J, and a positive or non-negative, better, sorry, non-negative, a non-zero coefficient of diagonal coefficient of this matrix basically implies that the, what happens at time t-j in region 2, for example, in this example, is going to affect what happens at time t in region 1. Okay, so we're going to call these matrices connectivities, basically between brain regions for each lag, because they describe the association between the measurements at different time points. Questions at this regard? At this level, I haven't yet described the experiment. It can be used both for resting state or task-based. Resting state, just for everybody, is the type of experiment where someone is put in the scanner and just let rest there, right? Left to rest there, and just you know, the activity is recorded. It's a spontaneous activity of the brain that is recorded, right? Okay, so let's describe. So if this is okay, so let's move on. So what we're actually interested in this case is the inference on these matrices here. One issue with this representation is, of course, the following. We are basically assuming that the association characterized by the matrices A are the same over the entire course of experiment. Of course, this is not completely adequate representation. We all know that the brain is not static, it's dynamic. So as I'm here talking with you, I have probably different connections than if I were here in the room or if I were at a movie theater, right? So there is, during the course of the experiment, when you look at different types of images, the expectation that your connectivities may change. So, and indeed, in the neuroimaging literature, there's been a realization that the nature of the connectivities is dynamic and fluctuating. So the connectivities may not be stationary over time. So instead of just considering a model for effective connectivity, we should better talk about a model for dynamic effective connectivity. How are we going to do that? Well, easy. We just put a T in those matrices, right? Very easy. of course, is not that easy eventually to understand the major concerns related to this. First, of course, if you put a T there, we have to understand that there will be transitions between different connectivity states. So we want to try, the major challenge here will be to try to model the transitions between these connectivity states. And this is particularly challenging with fMRI data, also because the data are by nature quite noisy. The second point that we maybe want to consider is that we have, at this point, when we add a time component there, a lot of parameters to estimate. But in reality, either because we really think that the type of connectivities that are realized in each moment of the brain are characterized by small network properties, or simply because of interpretation, what we want to obtain eventually is a sort of sparse representation of the connectivity networks. So we would like to somehow impose some sort of regularization in those matrices. Of course, when we add a time component in there, we also, especially if we want to be Bayesians in here, right? We also have another problem, which is computational issues and scalability. Just talking about vector regressive model without even going time varying, right? But even more so going time varying, we have generally some issues to go and consider a dimension for yt greater than, I don't know, 10, 20, 30. Think about ROIs. An atlas of ROIs is about 100 elements, right? If you would like to go for a voxel-based analysis, we'll have 100,000. Of course, I'm saying this, but then in the graded analysis, I'm going to use 27. But but for other reasons. But in general, so what we want to try to understand here and to come up with is a model that is able to maintain computational feasibility, allow uncertain quantification in a Bayesian setup, when we use a non-trivial number of ROIs. And by non-trivial number, I mean 4, 5, 10, which is common to see in the neuroimaging literature. So our proposal is to build a computationally efficient, time-varying Bayesian VAR approach for modeling high-dimensional time series with sparse connectivity networks. How are we going to do about that? Well, let me provide you with a visual idea of a model before I go directly into the math. First, we want to model these matrices, A1T, A2T, APT. The first thing to do is we look at a Raj and think we can stack these matrices into a tensor, right? And we have a tensor where each one of the dimension is n by n times p. P is the lag, remember, of a vector autoregressive model. And once we have a tensor at time t, we look again at Raj and think about a paragraph decomposition. And we do a paragraph decomposition of this tensor. I'm sorry, Raj, if I try to make this more interactive. So what we have is basically a decomposition of a tensor through a sum of the product of different vectors. We recognize basically 3 vectors: alpha 1, 1 of dimension n, alpha 2, 1 of dimension n, and alpha 3, 1 of dimension p. Basically here, so what we get again is a decomposition that allows us to represent the n by p tensor that we have on the left through, in this example here, a rank 3 paragraph decomposition. In general, you can think about a rank H, capital H, paragraph decomposition. But in this example here, we have 3 times n plus p parameters, right? And this is, if n and p are large, this is going to be a huge dimension reduction. So when we look at this slide, and when we look at the different composition, I'd like you to think about the interpretation of these vectors for a second. So alpha 11 and alpha 21 are of dimension n. Their product can be seen, if you want, as base connectivities between and basically that are going to be added one after the other to represent the tensor. The last component is alpha 31, that basically is a multiplying factor. And the idea here, and we'll use this idea later, is by modulating alpha 31 and using some sort of sparse inducing prior on alpha 31, we may be able to shrink to zero this coefficient if the lag is not needed. So if higher lags are not needed. Okay, so as I said, we're going to have a dimension reduction in this case. Now, what's going to happen at time t plus 1? At time t plus 1, we do the same thing, right? We consider a tensor. But the idea here is when we go from time t to time t plus 1, some of the components that were active before may be taken out. So for example, at time t plus 1, one component may not be needed to describe the connectivity at time t plus 1. So we just exclude that component. At time t plus 2, what may happen is another component could be taken out of the picture. So what we have, in other words, is a time-varying, at most, rank H decomposition that is whose components are going to be activated or not according to the ability to fit the data, right, at each time point. So, as this is basically what I just said, a rank H parallel decomposition with components that are differently activated at different time points. From a mathematical formula perspective, what we have, as I said, is a decomposition of the original matrices through essentially a parallel decomposition via the use of base tensor matrices that are activated or not on the basis of a selection indicator at each time point. Go ahead, yes. Oh, okay, great. So the question is, how do you choose your rank age? So let me say, so there is a quick answer is given latent indicator gamma ht, you can choose the rank age as high as you want, right? And then some of the components may be put to 0, and so they're not going to be used. And we've seen example, remind me, during the simulation, where actually we show that to be the case. If a component is not active, if the truth is that there are 3 components and we fit with 4, one selection indicator will be 0 across the entire time. Of course, you can still use all the tools that you may consider for choosing the rank age in typical tensor analysis, right? And that's something that we'll discuss more at length in the paper, but yeah, that's the basic idea. There was another question?
I just want to make sure I understand correctly. So here, the base activity is essentially across all the times, they're always fixed. So the only thing that changes is the combinations that you're taking.
Say it again, sorry, I lost the first.
So the base connectivities, those
yeah.
because they're essentially fixed across all of
Perfect.
the time points, but what is changing is the coefficients.
Perfect, exactly. The idea, exactly, perfect, exactly. So the base connectivities are the same across time. What changes is which one comes in at the different time points.
I can just follow up.
Oh, go ahead.
Okay. I go.
To follow up.
Yeah, so I guess effectively at the end of the day you have piecewise constants for that A matrix or A tensor. So what you change is the combination of those bases.
Yes and no, in the sense that these are matrices, right? But yeah, okay, yes. What you have is essentially the sum of constant components across time.
So even like the next time is still where one like the base component change, right? So even if the next time one is still back, it's still where one the base component will change. So you probably need a very large edge.
Yes, and well, depends, right? I mean, actually, we have been lucky, not, I mean, with the type of data that we've used, we never went over 10. So, but yeah, it could be possible, right? If I got your question correctly.
Yeah.
Other questions?
For a fixed T, what kind of non-uniqueness you have on the right-hand side? And in the way of interpretation, how close or similar to those components in singular value composition they are?
Perfect. So if I understand your question, right, you're basically asking, are these identifiable, right? In other words, right? Correct. I have the next slide, but the basic idea is exactly this. So there is no hope, let's say, to put it this way, to identify alpha 1H, alpha 2H, and alpha 3H. There is some hope to identify the product of the components through the selection indicator that change over time. What we're really interested in, though, is eventually in the reconstruction of the matrix of coefficient, and those are identifiable. Does that answer your question? Perfect. Thank you. Other questions? Okay. So again, so we just have these. If you want a time varying rank H parallel decomposition, max rank max H, I think. And we basically obtain the different composition of the different base matrices at the different time points. Of course, if we were to have all the gamma HT equal to 1 for the different ages, H, we will get a rank H parallel decomposition, right? The second point is going to address what I just discussed with Puramad, and then we have the other point that I want to say is by doing this, we have to obtain a reduction of the number of parameters that is now linear in the observation size instead of quadratic. So, of course, all is nice, but eventually the big problem here, or the interest, will be to study the transition, the model that you have for the transition probabilities, for the transition selection, for the selection indicators. This is indeed one of the challenges that I mentioned at the beginning when I said, okay, let's go dynamic, right? Finding the transitions. These transitions are in this model simply obtained through the sequence of latent indicators. And in the neuroimaging literature, and I guess if I were to make to ask here around a model that could be used for transition, one of the immediate one that comes to mind is Edematous models, which has been used a lot. The only issue is that if you think about our case, we would have to estimate a Edematous model for each one of the components, right? So that basically means that we have, let's say, even for states, we have lots of parameters to estimate, right? So this is not efficient for us. So we just did something different. We just used a Heisenberg specification that basically retains the Markov dependence, but allows us to relate the activation at time t with the activation at times t minus 1 and t plus 1. So the probability of activation at time t is going to be obtained through basically the combination of two components and basically two parameters. One general sparsity component that just describe the general probability of activation at time t as a function of the parameter theta h, and a component here I miss a t minus 1 that relates the activation at time t with the nearby time points, with the idea that if the probability if the nearby time points are activated, the probability of activation at time t is going to be increased as well. So we are basically, instead of considering all the parameters that describe the transition probability matrix in Edematous model, we just have two parameters right now to describe the transitions, which is again an advantage in terms of computation. And also we are going to, well, it can be shown that this corresponds to a new discrete ARMA process. The interest of these is in terms of specification for theta h and kappa h to avoid or to help avoid the phase transition problem, which is an issue with Eisen prior. I'm not going to go through that, but it's something that we can discuss at the end in case. Then, another way in which we can obtain dimension reduction is by putting some of the elements in our representation of the rank H paraffin decomposition basically close to 0, so that we're basically not going to represent connectivities. So let me start with Anirban first, because you did that before.
So the hidden markup model, is this to induce, I assume, to induce some sort of smoothness across time in your
Yes, so you want to, let's say, if the idea is, which is debatable, right? Could be debatable. The idea is.
My question, like, is that an assumption you want to make here that so if you look at the tensor at time t and time t plus 1, so Sparse smooth transition among those?
Yes, so the idea here is that, and it is a assumption that is often made in neuroimaging. It's debatable, we can discuss that in a second, that if you're looking at the picture, right, at a particular time points, right, you may have some connectivity structure that is going to persist at least till the task is active, right? So that's basically the idea. It is debatable, of course, because also if you consider the Fmri data are not at the temporal resolution and they do not capture neuronal activity in detail, right? So you may have actually sharp changes between 2 seconds that go between the scanner measurements, right? Because it takes two seconds for the scanner to take an entire picture of the brain. Does that answer your question?
And then if there are maybe at some points smooth transitions in some places, sharp transitions, can you comment on the flexibility of the model, because you're using the same set of base sensors and the only temporal thing is the indicator,
the selection indicator.
So,
yes, so actually, so this is a good question. And one thing that I didn't mention about this model, but your question gives me the opportunity to mention it. So one reason when I was talking with other collaborators before, who are neuroscientists, about the type of models that are used in the neurosciences that are based, for example, on hidden Markov models. In particular, one collaborator of mine, Vince Calhoun, who is now at Georgia Tech, I think maybe Brani knows him. His point was, yeah, these models are okay because they get the different states, but the brain doesn't transition from one state to another, right? Like, you know, like that, because following our model. This type of representation, however, allows, if you think of this in a different way, thanks to the time varying indicators, what you have is basically 2 to the h, right, combination, but describe 2 to the h states, right? And what you are basically saying is that when you move to one state to the next, you actually can compose some parts of what you, when you move from one time to the other time, these may be obtained from the composition of different subcomponents of a state. Does that? So that's a thing that I liked in this approach. I don't know if that answers completely your question, but.
So I want to put an icing prior.
Mm-hmm.
So
Sorry.
yeah. So that was defined conditionally, right? So, but for the joint prior for all the time points, so there's a prior nobody constant.
Yes, we use the Mueller approach. Basically, we use, so that's correct. We estimate the parameters, right? And we use the Mueller 2006 approach to sample from the parameters. I will go fast on that because I see we don't have much time, but yeah, we take that into account. Is that your question?
Yeah.
Perfect. Okay, so as I was saying, in addition to the transition, we also want to obtain a sparse representation. And we're going to use sparse inducing priors to obtain sparse connectivity networks. And the idea here is we want to obtain a more interpretable estimation of the connectivity parameters. And we have basically two sets of parameters. One are the alpha 1 age, alpha 2 age that are of dimension n, and the other are the alpha 3 age with our dimension p to take care of the lags. So we treat the two differently. For the alpha 1 age and alpha 2 age, I feel like Raj should have been here today because I'm going to use actually his suggestion for in his paper on Bayesian tensor regression, where he uses a multi-way Dirichlet generalized double Pareto distribution. Basically, this is a shrinkage prior. I think that here it's the best place to present this prior because probably many of you are used to this, but just in case someone is not, I'm just going to say it's not easy the first time that you see, but I'm going to go through it in a second. So basically here we have that the alpha j h are the prior is represented as a normal prior and the a shrinkage prior, where the value of the variance components, which basically is obtained, so the variance obtained is a product of three variance components, basically determine if alpha jh is close to 0 or not. If it is close to 0, we can take it out of the model. There are three variance components here. One is a parameter tau, which is a global shrinkage parameter. If tau is close to 0, all the elements of basically the connectivity matrices are going to be close to 0. The second component is the phi h parameter, which is across the different components. If when phi h is close to 0, then the element of the vector alpha jh is close to 0. For a less than 1, what we're basically saying is that we promote sparsity. What I usually tell when I present these work in other places, I say think about a beta distribution with a less than 1, and so you have a sort of a horseshoe type, or the mass is going to be close to 0 or 1. The last component is a matrix WJH, which is diagonal. And here the idea is we follow completely Raj approach. We use this is wrong. I changed this. I don't know why it stayed. It is not a Bayesian lasso. It's sort of the idea of a Bayesian lasso, but it's a generalized double Pareto prior. Okay. Anyway, it's approximately, you can think about a Bayesian lasso idea. That's not to go much into the details. I'm wondering what else didn't change from yesterday. Okay. Dropbox usually doesn't sync well lately. Anyway, sorry. Okay. So this is so in other words, we are going to have a sparse representation of a base connectivities. The other point that we want, the other vector we want to consider is the tensor margin corresponding to the lags. And here we want to use a prior that penalizes higher order lags. Basically, we want to assign growing mass to a neighborhood of 0 as the model complexity grows. How do we do that? Well, in first approximation, what we do is exactly the same idea as I just presented, right? So we have a normal prior with different components. Tau and phi h are exactly as before. What is going to change for us is W of 3h. And the idea here is that with high probability, the elements of W3H shrink progressively to 0 as we progress through the elements of alpha 3H. So to do this in practice, what we need to do is to introduce a lag indicator by which if the lag indicator is equal to 0, what we get is that the element, the diagonal element, is going to be taken from an inverse gamma, so different from 0, with, far from 0 for high probability. If alpha hk is equal to 1, then the corresponding lag is basically set close to 0. Not exactly 0 to allow the usual matrix inversions and work. So in other words, as we progress toward along the diagonal, we want eventually to hit 1, okay, from that point on. How do we do that? We use an idea that was presented for factor models by Legramanti, Durante, and Danson in a recent paper that appeared in Biometrica. And the idea here is just to consider the probability that zk is equal to 1 as increasing with the lags. And just to give a simple representation, right, we can consider a unitary stick, a stick of length 1. And the idea here is, okay, let's consider lag 1. And for a probability that zk is equal to 1, so it is excluded from the model for lag 1, is obtained through basically a beta random variable. So this beta random variable is going to chop off a piece of the stick, right? And what we are left is the remainder of the probability that ZH1 is equal to 0. So the lag is in the model. Now, when we go to the next lag, we want to increase this probability, right? So we take another beta random variable, and we combine them using what in Bayesian parameters is considered stable reconstruction, and basically we increase, by doing this, the probability that we're going to hit 0 at for the second lag, and we still go on along the diagonal. At the end, you can see the probability of getting a value different than 0 is going to shrink as we proceed. So this is basically the idea. I'm gonna go fast. I see only 10 minutes, but I want to show some of examples. So I'm not gonna discuss the posterior inference. It's pretty straightforward. And Jan just mentioned there is some issues with the sampling of theta age and kappa age, which are anyway solved using standard methods in the literature by now. So simulation studies, here I'm going to just show some simple simulation studies that we have done to compare our methods with others. For the purpose of this presentation, I'm just going to consider the comparison with the TVReg package in R, which is easy to obtain. The package, the model there uses kernel smoothing techniques to estimate the VAR coefficients. We consider different dimensions, 10, 40, 100 regions of interest. Notice that I'm not going to show results for n equal to 100 because we don't have any comparison. The TVReg approach is not able to handle 100 regions. Our model is, and does quite well anyway. So, but that's for another discussion. Just to keep things straight, streamlined, we are gonna, our truth includes a lag order p equal to 3. We use p equal to 4 in our model fitting to see if we're able to get 3. For these examples, we do some sensitivity analysis in the paper, especially in the revised paper. That's going to be up soon on archive, and then we have h equal to 3 as the truth, and h equal to 4 in reality, in our model fitting. We do well in the estimation of the tensor components, but even more so in the VAR coefficient matrices, especially in terms of detecting the true zeros, the sparsity based on the coefficient matrices. So looks like quite okay from that perspective. We also looked at the ability to find the transitions in multiple simulations, and we have at least in this case n equal to 110, good sensitivity and specificity. Things get better for n equal to 40, which is the next point. So the only thing that changes here for this presentation is n equal to 40 instead of 10. And we have different patterns, again, the same type of analysis, results. Okay, so this is an interesting figure, I think. So this is the truth. These are different intervals of time, 0, 100, 100, 150, 150, 200, 200, and 300. The columns are the different lags matrices, and we basically describe changes in the lag matrices across time. Estimation, this is just the posterior mean. We do well in terms of the comparison with truth. What I actually am more interested in this case is actually pointing attention to the lag 4, which is basically empty, right?
So I'll just one question. What is the signal to noise ratio?
I don't remember exactly. We did, so this is one of those that we did, we changed the signal to noise ratio. The reviewer also asked us to look at multiple sensitivity analysis. Exactly. I'm starting to wonder. Yeah, so overall, in the comparison, we did better. But this is mostly to show how it works, right? Typically, I mean, yeah, it was easy to find, but there was no, we see it, but also with analysis. But yeah, it's a good point. Okay, what else? Oh, okay, this is goes back to a question that I got before. One of the components is always 0, and we can retrieve it here, right? So maybe we're lucky with this. Maybe I showed you the, but seems to you something reasonable. That's what we're wondering and worried about. Let's look at the data analysis. I like this one because it's a reporter. And the idea here is the subject is in the scanner and reads words. I think it's 12 words per second of Harry Potter on the screen. So, and of course there is talking with a neuroscientist, for example, he had some issues with the experimental setup as well. But I mean, the ones that did this experiment did use this idea of using 12 words shown successively on the screen. Okay, so this is chapter 9 of Harry Potter and the Sorcerer's Stone. If you know Harry Potter well, this is a chapter full of episodes, twist plots. It's the first time that Harry actually flies, and that's actually one of the topical moments of the chapter that we're going to analyze. We only use 27 ROIs that were taken from a automated anatomical atlas of a total of 90 ROIs. These ROIs that we looked at were the ones that the neuroscientists have already screened as related with reading. Okay. Raj asked us to do course at the 90 and see the comparison between the 90 and 27 analysis. So it's something that we've been working on for the revision of the paper. But yeah, in this example, I'm showing 27. Raj, we get a strong reduction of known parameters, still a huge amount, 7,150, but if you think of the ones that we had initially, it's only 0.37%. And then further dimension reduction is achieved through the sparsity inducing priors. So this is a passage that was read, and we actually are going to analyze these particular time points when these words are shown. Blood was pounding in his ears, he mounted the broom and kicked hard against the ground and hop his sword. So we expect some sort of evocative, you can see the evocative nature of the play. Here, let's see. I was able to make it. I didn't believe so, Judy. But so this is the type of inference that we have. So this is just I'm showing the matrices right now, right? So just two things that I want to mention. First, something moves, right? Especially lag 1, which is good to see for especially our model. Lag 2 is a bit less dynamic, if you want. And the other thing that I want to mention is lag 3, which is pretty much well static, but it basically is 0, right? And this is what we expect. We do not expect typically fMRI data to go over lag 2. Actually, there is the general idea in the community of neuroscientists that AR1 is enough. So that kind of seems to confirm that idea. Well, simply here, I'm not a neuroscientist, but Ivor Kriben, who has worked a lot with this data, was able to recognize typical areas that are related with readings and their connections. So, for example, with object and face recognition, visual world, and we did also analysis, subject by subject analysis, showing some recurrent patterns, but also some heterogeneity. I'm not going to go much into the details here, but if you want to have more details, there should be hopefully soon. Soon means maybe still a month, a revised version of this paper online. So let's see, and wish us good luck. Concluding remarks here. So the first thing that I want to make is a general pitch. I think that the type of models that are being developed in the neuroimaging community right now are quite interesting. Also, you know, the problems that are tackled are interesting from statisticians. Why we do this eventually, this type of model, is to try to develop or obtain some neuro imaging biomarkers that can guide future interventions, right? Thinking about the difference in brain activity or brain connectivity, maybe the response, maybe the consequence or the symptom, better, of different propensities, for example, to stay on interventions, for example, to you know, fight or obesity or smoke propensity, smoking, right? So the idea behind this is that by looking at the different types of characteristics of the brain dynamics, right, one can learn more about inter-subject differences. There are also complexities due to the nature of the data that makes them particularly feasible, particularly helpful, I mean, in terms of developing models that allow computational scalability, either through dimension reduction techniques, as we have developed in this paper, but also through approximate inference like variational Bayes. We can also incorporate available information, and this may come, for example, through the use of structural MRI data or instead individual covariates. This can be done, and but it is, of course, going to add complexity. And then I just did say the last part. In general, there is, I think, a good, this is a good field, I think, for statisticians. And again, I think it's a type of research field that I like, and I suggest others to get into. This is it. I think I am 1 minute and 12 seconds after, but okay.
Thank you so much, Michele, for this very nice and very engaging talk. Probably got some exercise done as well. Yes. Yeah, I'm going to open it up to the audience for some more questions.
It's a very nice talk. I'm just curious about one thing, that you use the icing prior for that OPCA. So what if you do not use that icing prior, and like how much your results would be changed?
I don't know actually, because we didn't try eventually to go with the Markov model in this case, which would have been the natural thing to try, mostly because it would have required a complete separate development of a code, right? So, but yeah, so I actually don't know. Probably, yeah, not much, but yeah, I don't know, maybe other issues, right?
Curious what you did with the dirichlet concentration parameter, the A.
Oh, it was small, like we were following, yeah, the usual ideas, right? I don't remember. I mean, it was constant, right? Small 0.01, I can say, but yeah, that level of
Yeah, so just my experience playing with like in the Dirichlet class prior, for
Yeah.
example, with the considered parameters, the sparsity you recover can be quite sensitive to how you use it.
Yeah.
So in our, that prior when we wrote the paper, we did it on a grid search basically.
Huh. I have to check actually. Sorry, you got me in a problem there. I have to ask Wei what he did exactly. But sorry about that. Yeah, I think we fixed it, and I think we fixed it at a small value. So yeah. Let me double check though. Yeah.
Are there any other questions?
Yes.
Like some identifiability.
Yes. Yes. So this goes back to the question before, right? There is so we have to be aware of a type of things that we're interested in estimating. So the question is there identifiability? Are there identifiability issues? So of course we do not hope to. We are not interested in estimating exactly, the inference on alpha 1 age, alpha 2 age, and alpha 3 age singularly, right? We can learn about the product through the interaction with the variable selection indicator to some level, but we're actually interested in what I've shown mostly during the simulation is indeed the inference on the variance covariance matrix, but basically the sum of the different components, and for that, of course, there is no identifiability problem. So basically, yeah, the latent variables here are essentially unidentifiable, but we're just interested in the sum of the different products.
So can you do some interpretation of the latent variables?
We wish we could, and we wish we could do it. The interpretation, so in terms of inference, you mean at the end, at the moment, we are not with these, especially with these. I've been very careful not to do it. Eventually, so in a follow-up work that we've been thinking of with size variation of this method, we are open to at least be able, and that's why I'm saying that through the interaction of the selection indicator in this case, or by assuming dynamics in a different way of the alpha 1 age, alpha 2 age, and alpha 3 age. But again, we're interested in the product, right? We hope to be able to let's say, create some sort of space of latent variables that corresponds to some cognitive state. In that case, the experiment is on mice that try to recollect, but it's still, let's say, in high water as a project. We're still working on it. So, but yeah, eventually we would like to, but in this project we were careful not to. Does that answer? If you have suggestions on how to do it, please. Let's talk more afterwards.
Are there any other questions for Michele?
I just have one more. Do you expect to see some structural correlation for neighboring ROIs? And is there any kind of mechanism in what you've shown here that can account for that?
Yeah, that's a great question. And in this, no, I would like to do it though. So yeah, in this model, no, but I'd like to do it. So, but yeah, so so the general thing is that it's something that I've been thinking about, but I haven't seen this done more basically. So I take it back. So in the VAR, sorry, Abhishek, in the VAR literature, right? Let's see, when we looked at, I didn't see much of this relationship with spatial relationship, right? But when we described and we did this paper here, we related a vector tracing model with the structural Fmri that contains spatial information. So we did that in there, but not in this one. Okay, thanks.
Any other questions? Well, if not, let's thank Michele.
Thank you.